Okay, so this is meiosis 1, and it's in prophase 1. In prophase 1, the chromosomes come together to form the homologous pairs, and while they're doing that, they're crossing over to form tetrads. And as they're crossing over, the separate genes are switching. They're recombining. So now it's going to go into metaphase. So the recombined chromosomes are going to pair together, and they're going to start connecting to the microtubule spindle. So these are the spindle fibers in metaphase 1 as they're connecting to the homologous pairs. So in this part of anaphase, the spindle fibers are pulling the different pairs apart to the opposite poles of the cell, like this. Keep in mind that during prophase 1, when the chromosomes are pairing, their independent assortment takes place. This means that when maternal and paternal cells chromosomes pair, they're not always left to right or right to left. The order of the two is random. This plays a big part into gene variation. Okay, so now telophase is taking place. So the cell is dividing into two cells. So you can see the cleavage furrow right here as it divides. So now as meiosis 2 starts, so does prophase 2. So in this one, the two different cells now separate. The sister chromatids now are just together. Okay, so now in metaphase 2, just like in metaphase and meiosis 1, the sister chromatids are being connected to the spindle fibers. Okay, so this is anaphase 2. Similar to anaphase 1 and meiosis 1, the spindle fibers begin to pull out, but this time, instead of them pulling out the pulling apart the pairs, they're simply pulling apart the actual chromosome itself, pulling out the sister chromatids. Okay, so this is telophase 2. This is the last step of meiosis, and this is where telophase is where the two cells that occurred from telophase 1 form into 4 different haploid cells. So as you can see here, they're forming a cleavage furrow, and then as they separate, they form into 2 totally different cells. So in the end product is 4 haploid cells. Okay, so this has to do with the mitosis versus meiosis question. So this one main similarity between the two is they're both a form of cellular reproduction. Mitosis is different because it only forms 2 diploid cells, and both of them are identical, while meiosis, it forms 4 haploid cells, which has gametes. And another big difference is mitosis, again, is identical. Meiosis's whole idea is to get as much gene variation as possible, and that's their differences. So meiosis has to do with gene diversity because when things such as crossing over and independent assortment happen, the genes turn, the genes cross over or they just split, which causes them to make different genes as end products. So humans, we have 23 pairs, and the equation to find how many different possibilities are is 2 to the power of chromosome pairs. So humans have 23, so we have a total of 8,003,088,608 possible variations. But for our project, we use 3 homologous pairs. So by the end, you can get 8 different variations. Pertaining to the question number 3, which meiotic division 1 or 2 is more like mitosis? We think that meiosis 1 is because the final products of meiosis 1, like mitosis, still have their sister chromatids, the chromosomes. So pertaining to question number 4, what are some possible errors? We think non-disjunction is one error, and that is what happens when the spindle fibers are pulling out. maybe not just one from the homologous pair or the sister chromatid is getting pulled out, maybe both of them are, so there ends up being not an even number of sister chromatids on each side of the cell.
This can also lead to genetic diseases involving having too many or not enough chromosomes, such as Down syndrome.